Hi, my name is Alex. I'm a PhD student at UCSF in the Abbasi Lab, and today I'm going to be talking to you about my thesis project, which is generously supported by the ARCS Foundation. What I'm interested in my project is to understand what can we learn from parts list of the brain, and secondarily, how can we put these parts together to understand more about the complex function of the brain. To give some background, what we're interested in is data that has been recently generated using new technologies from our collaborators at the Allen Institute for Brain Science to molecularly fingerprint millions of cells at once in the mouse brain. What I'm showing you on the left is a sample of that data, where each of these little pieces is one section of the mouse brain, moving from the front in the top left to the back in the bottom right. Each of these little sections is made up of millions of small dots, and each dot is one cell, and they're colored by different types of cells that we've been able to identify using previous knowledge. So you can see that there's already, in just by your eye, a lot of detail that you can identify where different types of cells are clustered in different areas. And when I say molecularly fingerprint, what I mean is that we measure hundreds of molecules in each of these cells at once. On the left now, I'm showing, for example, one that's related to sleep regulation, where a dark color means that that cell uses a lot of this gene, and the light color means that that cell uses a relatively little. Here's another one, the dopamine receptor, which hopefully you'll be familiar with, which is related to regulation of learning and motivation. And you can see that cells that make this gene are really clustered in a couple areas, mostly in the front of the brain in the top part of the diagram. But no cell stands alone. And what we're really interested in is how can we find the groups of cells that evolution has put in place or in specific places for specific jobs, and then hopefully to understand how those positions and how those groupings of cells give rise to complex functions. To do that, we lean on a machine learning solution using the same types of technology that underlies ChatGPT, what is called the transformer neural network. And using this method, we establish one of the newest and most detailed accounts of the different subregions in the brain and the spatial arrangement of cells inside them. This is an example of what comes out of our method. On the left, I'm showing you a cartoon that was also generated by collaborators at the Allen Institute, which is to the sort of sum of human knowledge so far, the different types of regions in the brain, where each subregion, for example, like the dark green color, is the sensory area of the mouse brain, gives a different subregion as we know it. What's showing on the left took maybe 5 years of effort and hundreds of different experiments to generate enough data and to have people look at manually the different types to understand where these regions start and end. But on the right, what I'm showing you is what comes out of our method, where now I'm coloring each cell by the different subregion that our method detects in a fully automated way with only one mouse. And you can see that we get a lot of the same structures, and we even get some sort of more granular and newer structures that we're excited to explore. We're excited about potential diverse applications in areas such as disease, for example, understanding the roots of psychiatric or neurodegenerative disease, or, for example, to understand the ways that the brain transforms data into information and makes decisions. Because if we could understand where the different cells are and how they work together, maybe we could understand how they create the kind of function that we're interested in understanding mammalian function. I just wanted to say at the end, thank you so much to the ARCS Foundation for supporting my thesis and supporting the innovative research that's going on all across different universities.